Mode.
Hello, good morning, good afternoon. Welcome to today's GastroPlus User Group webinar. My name is John Brown, and I'm going to be acting as the moderator for today's session. And it gives me great pleasure to be able to introduce Wei Zhu and Bingfeng Zhao, who will be presenting today's session. They're both from Merck in West Point, Pennsylvania, and they're going to be sharing with us their work on the applications of customized GastroPlus ACAT models to address challenges in formulation development. So we'll have two presenters. The format that we'll take is each presenter will present on some case studies, and then we'll save our questions to the end of the session. In terms of the audience asking questions, that's something that we always welcome on these webinars. We welcome the opportunity for that discussion. And we'd ask you to present your questions actually through the GoToWebinar software using the questions box in the software panel. So if you submit the questions as we go along, and at the end of the presentations, we can actually read out those questions and have some input from our panelists to help address any queries or any comments that have come up during the session. The other thing I just need to remind everybody of is, as in all of our GastroPlus webinars, we like to record the session, and then the recording is placed on the Simulations Plus website for people to refer back to in the future. Of course, in case of colleagues who've not been able to make today's webinar, the webinars are always accessible from that point. So, without further ado, I'm going to hand over to Wei and Bingfeng, and we very much look forward to today's presentations. Wei?
Thank you, John, for the kind introduction. Hello, everyone. My name is Wei Zhu from Biopharmaceutics, Pharmaceutical Sciences and Clinical Supply in Merck Research Laboratory. Today, we will share how we use GastroPlus in our group to provide inputs and guidance from biopharmaceutical perspective during various stages of drug development in Merck. Specifically, we will present several case studies to demonstrate how we use customized ACAT models to address the challenge during the formulation development. So the slides, as John has mentioned, will be co-presented by Bingfeng and me, as I will present the first two cases, and Bingfeng will present the last two cases. We will cover the GastroPlus work in the following areas. The first one is the prediction of drug-drug interaction with acid-reducing agents. Second one is the lower gut regional absorption and control release formulation evaluation. Third one is the physiologically-based absorption modeling as an alternative IVIVC approach. And the last but not the least is the oral cavity compartmental absorption model for assessment of sublingual administration. As John mentioned, we can take questions, comments together at the end of the presentation, if there's any. Let's start with the first part, which is using physiologically-based absorption model as an alternative approach to assess Achlorhydric effect. Achlorhydria or hypochlorhydria is a condition commonly known as a lack of or reduced secretion of gastric acid in the stomach, which has been shown to significantly impair the absorption of weakly basic drugs, which show low solubility at high pH or intestinal pH range. The change in gastric pH could be due to several reasons, such as using of medications such as PPIs or H2 receptor antagonists or antacids. it can be also associated with different diseases such as gastric mucosa infections, HIV infections, stomach cancer, etc. Gastric acidity has also been shown to vary with different ages and affinities. For example, elderly Japanese has shown higher gastric pH compared with the elderly North Americans, and the elderly North Americans overall has high incidence of higher gastric pH versus the younger group. There are several publications show that weekly basic drugs such as dipridomyl ketoconazole has low and variable absorption, which is thought to be preliminary due to slow and incomplete dissolution of these drugs under high gastric conditions. This can consequently lead to subtherapeutic exposure, and in such circumstance, at least there will be a concern on product label if the Achlorhydria effect persists in the final market formulation. To overcome the Achlorhydria effect, one may need to develop an alternative or an enabling formulation to mitigate the Achlorhydria effect, such as by using solid dosage form containing organic acid to enhance dissolution by increasing the microenvironmental pH, using solid dispersion to improve the solubility at high pH, or using different API forms such as salt form of weakly basic compounds to mitigate the slow dissolution in Achlorhydria condition. Within Merck, we have been utilizing biopharmaceutical tools to evaluate Achlorhydria effect. The in vitro evaluation in the form of either solubility or dissolution experiments is an important and often the first step of the formulation evaluation. We also use the dog model, which is the most common preclinical animal model for formulation screening. To avoid variability and generally low gastric acid output in the fasted state compared to the humans, we use pretreatment with pentagastrin to mimic the normal fasted state in humans, and pretreatment with an H2 blocker, most typically famotidine, to mimic the Achlorhydria state. The absorption modeling work on the evaluation of Achlorhydria effect has been implemented throughout the drug development stage in Merck. At early stage of development, absorption modeling could be utilized to map the anticipated behavior of the compound as a function of stomach pH to predict any bioavailability risk in clinic, and inform whether further formulation investment is warranted. Once the risk is identified and formulation development is initiated, the absorption modeling could be used to either help translate the in vitro data to an in vivo response, which so-called PBPK modeling based IVIVC, or help translate data from animal study to the clinic. An earlier work that was published in 2011 by our group on investigation of Achlorhydria effect of a Merck investigational weekly basic compound incorporated both in vitro dissolution data and dog PK data into the models to project clinical exposures. The base model was developed in GastroPlus and accurately captured the impact of stomach pH on exposure as seen in the first-in-human clinical studies with conventional formulation. Once the base model was established, the observed dissolution data, which was taking the fraction released at 15 minutes of each dissolution profile, were used as input in the calculation for the in vivo stomach solubilization, and subsequently as the major driving force for absorption. However, it was a hybrid model, and some important assumptions were taken with this model, such as a separate stomach and intestinal solubilization processes, and the assumption of no in vivo precipitation, which represents the limitation of the application of such model to other compounds. As we continue this journey on developing and optimizing the biopharmaceutical tools for Achlorhydria evaluation, we have used the PBPK-based absorption model incorporated with in vitro dissolution data of low buffer capacity citrate buffer to represent the achlorhydric conditions and tested our in vitro in silico models with several in-house and market compounds. In this screen, I use a Merck compound A as an example. So this is a weekly basic BCS2 compound, and it has a steep pH solubility profile with pK of 5.7. And as you can expect, the low solubility in the intestinal pH range may have an impact on drug solubilization and absorption in achlorhydric state. Here we had used the dissolution model to look at the in vitro release of conventional formulation tablet 1 in both SGF and pH 3.3 citrate buffer, and we found a significant difference on the dissolution of the drug. The dissolution for tablet 1 has no profile, especially in the pH 3.3 media, so we applied the kinetic solubility value in the GastroPlus after we set up the baseline model with clinical data in fasted state with normal stomach. So it turns out the model predicts the exposure in the achlorhydric condition was largely in line with what we observed in PPI co-dosing arm of the study for tablet 1, as you can see on the right side of the slide. And as you can see, the simulated AUC and Cmax ratio with and without PPI co-dosing are in agreement with what we observed in clinic at 40 milligram dose. Once we have the baseline model for conventional formulation and pointed out the risk, the formulation team developed several alternative formulations with the aim of overcoming the achlorhydric effect. And let's take one of the enabling formulation, tablet 2, as an example. Our group did several a priori predictions for this before we see the clinical data. So we incorporate the dissolution profile either by using an artificial API particle size setting or using the GastroPlus Z-factor models of the enabling formulation in the two media to the baseline absorption models and predicted a 60% relative exposure for the enabling formulation in PPI co-dosing condition versus the normal condition. So we can also do a prediction of achlorhydric effect in different doses. And in this case, we did at 20 to 80 milligram doses and predicted a largely similar percent loss of exposure across the doses in the case. So what we also do within our group is oftentimes we challenge our model with sensitivity and scenario analysis. In this case, since this is a weekly basic compound, so even though we can use various in vitro tools, it is still usually challenging to accurately predict the in vivo precipitation of the compound. So what we do is to look at the sensitivity of the in vivo precipitation on the prediction of achlorhydric effect, which would be a ratio here. So which is on the top right side of the slide. Because of the possibility of the possible biomedical effect resulting in lower drug diffusivity, although the in vitro data, I have to say that suggested it is less likely the scenario for this compound. A sensitivity analysis on the diffusion coefficient, nevertheless, was also investigated as shown on the bottom left side of the slide. So when we have the clinical data back, we compare the observed data versus the model predicted outcome. As you can see in the bottom right side of the slide, so the model seems to be over-predicting the achlorhydric effect by about 25% to 30%-ish for this compound. But however, overall, if you look at the whole model development process, the model actually has accurately captured a reduced bioavailability of the conventional formulation, but an improved bioavailability of enabling formulations, although not to the extent that we have seen in clinic. So in the condition of co-administration with PPI. So the model at least pointed us to the right direction as we advance this enabling formulation, which is tablet 2, to the clinical study. So overall, this case study demonstrates the utility of the absorption simulations in assessing the PPI effect on clinical bioperformance of various formulations, which is also helpful to get further prototype formulation development. So I'm going to switch to the second part of this presentation, which is the application of the absorption modeling for estimation of lower gut absorption to support CR feasibility assessment. So there are several factors dictating the PK profiles of a CR formulation. The release rate of the formulation, which is largely considered a property of the formulation. The regional bioavailability in the intestine, particularly in the lower gut, is compound dependent and can have significant impact on the pharmacokinetic profile. And the systemic pharmacokinetic parameters or relative rates of input and elimination dictates the overall pharmacokinetic profile. So there are also several special considerations on the lower gut absorption compared to the small intestinal absorption. For example, the solubilization capacity in lower gut is generally lower due to low luminal volume in colon and also low bile salt concentration presence. And the regional dependent absorption across the GI tract needs to be taken into account, as the permeability difference is expected due to tight junction in lower gut compared to the upper GI, and the regional dependent transport expression and the efflux need to be taken into account as well. So in order to evaluate the CR formulation, the in vitro release study is often the first step. The dog model has been widely used as many examples were reported in the literature to evaluate the PK of human dosage forms. So generally, the dog model has good predictions for the osmotic tablets, but for matrix tablets, the predictability usually depends on the mechanism of either erosion-based or diffusion-based mechanisms. And the difference between species needs to be considered, such as the stomach contractions, hydrodynamic flows, GI transit time, GI fluid volume, metabolism, and transport. So from the in silico side, based on the in-house experiences of in vitro cell permeability and correlation with dog colonic bioavailability database, we can use it as an early screening tool, but certainly need to consider the difference of in vitro Heco2 cell models versus the in vivo system, such as the difference on the enzyme transporter expression levels, the difference of the tight junction between the cells and the in vivo conditions. So here in this presentation, we will share how we use the modified ACAT model to estimate the lower gut absorption and further translate the absorption model from dog to human model for assessment of CR formulation. So this is a general workflow of development of this lower gut absorption model in GastroPlus. So the first step is to build a oral absorption model based on dog IV, PO, PK data by using GastroPlus PK Plus or Phoenix Winner Lint for compartmental PK analysis and estimating the PK parameters. Once we have the baseline model, the baseline oral model, we can calibrate the lower gut permeability settings in dog model against dog PK data of colonic dose formulation, mostly solution formulation, by adjusting the ASF settings for different colonic absorption model to fit dog colonic dose PK data. Then we validate the permeability settings in dog model against the PK data of orally dosed control release formulation. And afterwards, we extrapolate the lower gut permeability settings in GastroPlus from the dog model to human model using the ASF scalar, which I'll describe in the slide, in next slide. And after that, we can predict the bioperformance of the CR formulation in clinic with the calibrated human model. So here, this is the case study. I think I'll just use a case example which is a Merck compound B to demonstrate the in silico work process. So this is the BCS2 compound with decent solubility in buffer or passive. However, prior to first-in-human study, because of the projected low half-life and the C12 targets with relatively narrow therapeutic window, there is a desire of developing a CR formulation. So the good news is the regional absorption study via colonic dosing has shown a good relative bioavailability for colonic dose versus the orally dosed solution formulation in dog model. So as I described previously, the first step of the workflow is to develop the CR model in GastroPlus, was to build an oral PK model in dogs. After we have the oral PK model ready, as you see in the step 1 here, we modify the default dog model in GastroPlus to reflect the condition of colonic dosing of solution formulation, either as a 1-compartment or a 2-compartment lower gut model. As you can see, the transit time and the volume has all been set to 0 for stomach and small intestinal compartments. So the 2-compartment model is the default dog theoretical SAV model in GastroPlus, which, while in the 1-compartment model, the transit time were added by the cecum and colon transit time in the default setting of the 2-compartment model. So the default ASF setting in 1-compartment model were adjusted accordingly based on the definition of the ASF in theoretical SAV model, which take into considerations of the SEF in ascending colon, SEF in jejunum 1 compartment, and the radius of the ascending colon. Once we have this baseline model set up, we optimize both Model 1 and Model 2 against the dog PK data with clonically dosed formulation as shown here in step 2 by adjusting the lower gut ASF values in each model. We obtain the ASF scalar that I mentioned previously for human model in step 3 for both 1-compartment model and 2-compartment models by using the ratio calculated from optimized dog ASF against the default dog ASF. So let's look at the actual work for compound B. So here I didn't show the dog oral PK model, but showed the more critical data of the models, which is the chronically dosed dog PK data. As you can see, the optimized 1-compartment and 2-compartment models showed acceptable fittings against observed data by adjusting the lower gut ASFs in each model. The total fraction absorbed in both 1-compartment model and 2-compartment models are generally comparable in both models. As we move on to validated model with the orally dosed CR formulation dogs, as you can see, on the left side is the in vitro release profile of the CR formulation, which we applied to the absorption model. And on the right side are the simulated versus the observed PK data for the CR formulation in dogs. As you can see, those absorption models has acceptable fit to the observed oral PK data for CR formulation. The model with 1-compartment in colon predicted a higher FA than the model with the 2-compartment model. But overall, the good fitting of the data brings us more confidence to extrapolate this model basically before we move on to predict the outcome in the human. So to set up the human absorption model in GastroPlus, we modify the default human model in GastroPlus by using the previously obtained ASF scalar, as I described 2 slides ago, which was in the step 3, based on the ratio of the optimized dog ASF against the default dog ASF. in both one compartment model and two compartment model. And we extrapolated into the humans for the lower gut ASF settings. So prior to that, similar to the dog model, the two compartment model is the default human theoretical SAV model in GastroPlus, while in the one compartment model, the transit time were added by the cecum and colon transit time together in the default settings of the two compartment model. Similarly, the default ASF settings in one compartment model were adjusted accordingly because based on the definition of the ASF in theoretical SAV model, which takes into consideration of the SEF of ascending colon, SEF of jejunum one compartment, and the radius of the ascending colon. So we then modify the default human model in GastroPlus by using these previous obtained ASF scalar, and we are ready for the prediction. So here is the prediction of the clinical bioperformance for the 3 CR tablets with different release rate. On the left side is the in vitro release of the formulations, and on the right side are the simulated PK profiles in both one compartment and two compartment models at 2 mg dose. The predicted AUC is generally in line with observed data, although for confidential reasons, I cannot show the data here. So I think this is the end of my part of the presentation, and I'm going to hand over to Bingfen to present the last 2 case studies on PBPK modeling based IVIVC and absorption modeling of intraoral absorption. After that, we can take questions and comments at the end of the presentation. Thank you.
Thanks, Wei. And again, my name is Bingfen from Biopharm, same group with Wei. So I will continue our presentation with the next 2 case examples. So the first example is the physiologically-based absorption models as an alternative approach to build IVIVC. So the IVIVC here stands for the in vitro in vivo correlation. So this work has been already published recently in AAPS journal. And first let's define what is IVIVC. So IVIVC is a predictive mathematical model to find a correlation or relationship between in vitro performance. Usually the in vitro performance is the rate or extent of drug dissolution or release from an in vitro study, and in vivo release of drug and formulation, for example, the plasma concentration profile, and also maybe the amount of drug absorbed. So there are several classes of IVIVC, class A and B and C. The level A IVIVC model actually describes the relationship between the entire in vitro dissolution time course and the entire in vivo response time course, which because it can include the entire dissolution profile and the entire response time course as the PK profile. The level A IVIVC is basically recommended by the regulatory agency, but also it's pretty complicated because you have to match the whole entire profile. So there's several ways we will do IVIVC model. The most traditional way IVIVC approach is called deconvolution and convolution IVIVC model. So as you see the figure at the bottom, first of all, you have to deconvolute a set of pharmacokinetic data to generate an in vivo absorption profile, and then combine with the in vitro release profile and build a mathematical model for the correlation between the in vitro fraction release versus the in vivo fraction absorption. And then if the model can be successfully built, if you have any new in vitro release profile, you can using this correlation to estimate the in vivo absorption profile, then convert back to the pharmacokinetic profile for these new formulations. But in general, this approach will be easy if the whole entire profile has a linear relationship, and then in this case it will be pretty easy to generate such kind of IVIVC model. But very often that you will see that the correlation may not follow a linear trend. So for the case example, I will discuss later on this. We're trying to using this conversion approach to generate IVIVC. However, we have to end up using two stages of linear relationship. So which means that we don't have a very nice linear correlations from the whole time period. So we have to split it into two stages of the model. So that would be more complicated to be approached. So that's why we think about whether we can use some alternative approach to complete the IVIVC for this compound. And so, I mean, recently, as you know, that the physiological-based PBPK model has been used quite a lot. And I got this flow diagram from the paper published in 2011, published by Xingyuan Zhang from FDA. And she actually summarized a very nice flow diagram of these physiological-based absorption models and listed this approach. So basically, from this flow chart, you can see that the first step will be trying to construct a PK model or construct the drug information. For the PK model, is basically just trying to collect the pharmacokinetic profile of parameters based on the available IV data or oral data with fast dissolving formulations. And they also need a drug information, basically is the formulation information, physical chemical properties, and etc. information. So with the drug information and PK model combined, you will be able to develop models, PBPK absorption models, but you probably also need to spend more time on the next two steps, which is to optimize the model, to verify the model and refine the model. So because you have, so basically the PBPK model is a bottom-up approach. So you have to incorporate lots of parameters, but not every parameter you have high confidence. So you definitely need to optimize those parameters with high uncertainty. And also the model should be able to fit PK data well, but not only one PK data set, but also different PK data sets, including like different dosing regimen or different formulation or whatever, different food condition, etc. So overall, based on these two steps, the model optimization and validations, what you got is the question is how predictive of your model is. So you have to build a confidence about your models, and based on your confidence of your models, you continue to think about how to use the model, what range the model will be predictive and it will be successful to address the questions we have. So that's why at bottom it will be the model exploration. So for IVIVC approach, if we're using the PBPK models, we are trying to follow the same flow diagrams and use the current model to achieve IVIVC model for the case examples, the case samples of our compound. So here is the compound we use. This is called compound C. This compound is actually from this slide you have see there's one fast dissolving formulation, which is IR formulation. We also have two control release formulations. One is the matrix-based formulation, which is erosion-based formulations, and the other is the multi-particular formulations, which is more like dispersed formulations. And in each category of control release formulation, you have three different release rates: fast, intermediate, and slow. And the compound itself, we have it has a dose of 4 milligrams. It's pretty low dose, and we have very decent solubilities across the pH range. So you will see that the dose number actually is way below 1. We're using LCTK1 as a cell line to measure the permeability. So overall, the compound is a highly soluble compound with moderate to high permeability. And so first of all, as I said, we need to build a PK model, and we also collect drug information. So PK model can be built just based on fit the IR PK data. So you see we have the green curve here stands for the immediate release formulations. So based on that PK data, we fit the pharmacokinetics using GastroPlus PK plus modules to get PK parameters, and then we will So this is the first step to build the models. So overall, that's the physiologically-based IVIVC modeling strategies. So first of all, we incorporate the compound C's PK parameters and also the related physical chemical parameters, those drug informations. And because we are trying to model the control release formulation, so we have to get input from those control release formulations. So we're using the GastroPlus Weibull functions to fit the individual dissolution profile and use the Weibull function profile as the input for those control release formulations. And similar strategy as I used, as Wei said in his chronic studies, in order to fit these data as well, we need to adjusting the absorption scaling factor, which is ASF value here. So ASF, again, is a multiplier accounting for the variation of surface, volume, and pH effects, and other absorption determining factors for the absorption rate. So basically, we're trying to do here is adjusting the absorption scaling factor, that factor to fit the pharmacokinetic data for each class of formulations. But because we are trying to fit the oral profile, so we have to adjust the ASF values for almost all of the compartments for the intestinal compartments, including duodenum, jejunum, ileum, and cecum colons. And for simplicity, we just select 5 ASF values. For duodenum, jejunum 1 and 2, we're using single ASF. We're using different ASF values, and we're using same ASF values for 3 ileum compartment and same ASF value for both cecum and colon compartment. So duodenum to jejunum, we actually accounting for the absorption region at upper small intestine, and ileum and cecum and colon are the compartment at lower small intestine and the lower GI tract. So again, first of all, we got the immediate dissolution profile from 2 different formulations, the matrix formulation and the multi-particulate formulations with different release rate. So here, the open rectangle here is our experiment data, and using the Weibull functions in the GastroPlus control release manual, you can find the Weibull functions model. You can using the model to fit the initial data to get a Weibull function curve, as you see here is the blue curve here. And you can also get the Weibull function parameters, which is A and B, and then using this Weibull input as our input for the model. So again, so this is also showing how to adjusting the ASF values for fitting the pharmacokinetic profile. So basically, if you remember that GastroPlus has the default physiological profile in its gut physiological tab, which is GastroPlus OPT log D models SAV 6.1. So you get these default ASF values from that model, and from the which showing here is the left-hand panel, and you see that the ASF values from duodenum, jejunum, ileum are pretty close, from 2.6 to 2.8. And I mean, we try this default model to fit data. Actually, this didn't work very well. So what we do here is you can actually switch to a user-defined model by switch the you can in the job box of that physiological tab, you can define it as a user-defined model. So basically, on the right-hand side is the user optimized model. So we optimize the ASF values using our first of all, we're using the manual to test initial estimates, and then we're using the GastroPlus optimization module to find optimization in the numbers. So we got a new set of ASF values here, and you can see at up GI, duodenum, jejunum, we got a higher ASF values compared to the default ASF values. But at the lower GI, we actually reduced ASF values to very low numbers. So what does this mean to me? I mean, this actually means that the drug absorption is likely mainly contributed by up GI, and because we have to adjust higher ASF values to fit data, And also at lower GI, we are assuming that the drug absorption will be limited, which is also consistent with our chronic absorption study in dogs. We only found that 30% of drug can be absorbed in the chronic area. So that's the way we adjust ASF values. So this is only for matrix tablet, and we do the same thing for the multiparticulate tablet. However, I mean, I think that the adjusting of ASF factors is considering as an indication of physiological reasons for formulation behavior reasons. But also, it's probably difficult for us to directly point out what's the real mechanism of the reasons. I mean, it could be related to physiological impact, but also could be the behavior of formulations. But this approach actually gives you insight about the absorption pattern of those control formulations. So again, this is the predicted versus observed pharmacokinetics using our approach. The matrix formulation and multiparticulate formulations and the observed data and simulated curve are shown in this slide. And basically, you can see that initially the absorption is pretty fast, but the plasma concentration drops pretty quickly and reach a kind of like a flattened area from 6 hours to 24 hours, which may indicate that the absorption rate goes slower. So that's why we also set a lower ASF value to account for the slow absorption rate and the lower GI. So for IVIVC model, it's not only fitting the PK parameters, also we need to check the point estimate and deviations, which is required by the guidance of regulatory agency. So in general, we are looking at a deviation of prediction within 15%, within plus and minus 15%. So here is the overview of the predicted value and also the deviations. So PE here stands for the prediction error. I mean, so here we see most of the number here is below plus minus 50%. Only the matrix 16 hours is a little bit over that criteria. So I think the model is pretty successful if we using a different set of ASF values to predict different control rate formulations. But ideally, it would be good to using a single set of ASF values to predict both matrix and multiparticulate formulations. We also try that approach, but we see larger deviations, particularly for slow release formulations. And you know, different formations could have different region absorption patterns, which will be different from different formulations. So in this case, we are still trying to say we're just using a different ASF set for different formulations as our IVIVC approach. So, so this is the third case example. So next example is to discuss the development of oral cavity compartment absorption and trend model. And this is a model also called OCCAT model. I think this model has been released, should be available in the new version of GastroPlus version 9. And this work is also collaboration between Simulations Plus and Merck back to several years ago. So the intra-oral delivery actually is an alternative administration approach that aims to deliver a drug substance via oral cavities. So we don't really have a physiological-based models. I mean, you probably will feel that in GastroPlus we have oral cavity compartment, but it's not like fully mechanism-based and physiological-based model. So that the work is trying to develop a fully physiological-based model. And we also had the publication of this work in early 2015. So let's just go through this schematic diagram of the OCCAT models. So here in this slide showing schematic show of the compartment OCCAT compartment models. So the boxes here represent the individual oral cavity compartment, and the blue arrow actually is the drug exchange between the compartment to the perfusion layer. And also in oral cavity, you know, we also secretes salivas, and the orange arrow actually shows the saliva secretion from buccal and flow to gingiva and other tissue compartments. And the orange dashed line actually represents the transfer of API upon swallowing, because the model actually said as the swallowing will be triggered when the saliva accumulates to certain levels, and it will trigger swallowing, and the dashed line actually represents the swallowing of API. So again, sorry, the model actually, when drug got swallowed, you can see that also the model connected to ACAT models, because all of the swallowed API will be getting into the GI tract. Then we will be using the ACAT model for the further drug absorption. So this model also considers the anatomy of oral mucosa. So the anatomy of oral mucosa has two major layers: the oral epithelium and the lamina propria. So for each layer, we define as different sublayers to reflect different cell layers in the anatomic situations. And then, so from here, we using these two layers, epithelium and the lamina propria. And for each layers, we also divide it by different sublayers. And also the model accounts for the drug dissolution precipitation in the saliva, which is in the oral cavities, and also consider the swallowing of dissolved and undissolved drug. So the absorption of the dissolved drug from saliva is via instant equilibrium of unbound drug concentration at interface between saliva and epithelium. So the P here represents a partition coefficient. It's not a permeability, but a partition coefficient. And so it's an instant equilibrium reached at any time. And so if you can imagine that if the drug was swallowed, the concentration in saliva will be reduced instantly, and the partition will change instantly as well. And in this case, you will see that the drug will also diffuse back from epithelium layer to the oral cavity in the saliva. So basically, what I'm trying to say is the accumulated absorption in oral cavity model will not always increase. Sometimes it will decrease because of back diffusion. And the drug molecule within the epithelium cells will further diffuse deep into tissues. So that will be controlled by another parameter called diffusivity. So diffusivity represents the diffusion rate to deep tissues. And in the lamina, actually was well perfused by the blood vessels. So over there will be a systemic circulation uptake of the drug molecules. And also we have to consider the non-specific binding of drug in tissues. And the partitioning coefficient actually represents the total drug concentration versus the concentration in the saliva, but also equals to the total drug concentration in the tissues versus unbound drug in the epithelium cells, because you see that saliva concentration, the unbound saliva concentration should be equal to the unbound concentration in the epithelium cells in order to reach equilibrium. And all of the compartments was actually, we got different tissue parameters based on lots of literature search. For example, like thickness, surface area, blood flow, and pH for each tissue compartment. So here, that diffusivity and the epithelium saliva partitioning coefficient is two key parameters for this model. As I said, the partitioning coefficient is also a kind of reflects the tissue unbound value, which is they have a kind of like relationship of reciprocal relationship. And we actually in this slide we actually tested about 10 model compounds, and we are using pig, buccal, and sublingual tissues, and to do an in vitro formulation assay. And based on the assay results, we are using the model to estimate the partitioning coefficient and diffusivity, and we build a correlation of diffusivity and FUT or partitioning coefficient with the log D value. Log D value actually is a physical chemical property. So the reason to do that is trying to build kind of generic equations whether we can estimate these two key parameters using physical chemical property of a compound like log D values. So this is the equation already available in the programs. You can use it. Of course, you can also optimize these two numbers if you have some preclinical data. So let's go to the example of intramesal sublingual tablet. So intramesal sublingual tablet is a Zolpidem tartrate compound, which I listed this drug information here. It's also well dissolved, and it has and I got the PK parameters from the IVIVC data of Zolpidem, and this study actually assuming a holding time of 2 minutes. The pH in the oral cavity was set at 5 because we actually did this drug is pretty formation is a little bit acidic, so that's why the pH in oral cavity is reduced by 2 units. So the modeling approach for this study is we first need to use an ACAT model to simulate Zolpidem oral tablet, and then we using this physiologically-based multi-compartment oral cavity model, OCCAT model, to simulate Zolpidem Sublingual tablet. And for comparison, the traditional ACAT model also including this one-compartment oral cavity model. So we call this as a simple model. So for comparison, we're just using that simple model also to simulate a Zolpidem Sublingual tablet. And we're assuming that ASA value is in here from the duodenum ASA value. So here's the simulated result, which is the comparison between OCCAT model and ACAT models with oral cavities for Zolpidem Sublingual and oral tablet. So I mean, the fittings, you can, I just trying to highlight the early absorption fittings at the figures at the lower part. So here you see that the blue curve represents the simulated PKs of oral tablets, which you see the slightly delay of absorption, and the red dot and curve, which represent the simulated PK of intramesal tablet, the sublingual tablet, and you see that the model actually capture the rapid, more faster absorption using the sublingual tablet. And also we tried the simple model. The simple model actually was showing at a dot line, simulated a dot line, and basically the simple model actually didn't predict the observed data well for the sublingual tablet. So in this case, I think the performance of this OCCAT model is acceptable. It differentiates absorption rates between the sublingual tablet PK versus oral PK. It also kind of has a better predictions compared to simple models. And in addition to that, you can also get lots of diagnostic plots from these OCCAT models. So here you can actually find a lot of information there. There's a drug absorption and dissolution profile. On the left hand side, that's probably information you're pretty familiar in the ACAT model, because you have absorption dissolved, the amount absorbed, and also the total amount entered the system and circulation. But this pink line is also a new absorption profile, accumulate absorption from the intraoral side. So as I said, that because of back diffusion, it's not always increased, but also can decrease because of the swallowing, right? And you can also see the swallowing process from the figure on the top right. So you can see that the drug can be swallowed, and no matter both dissolved and undissolved drug amount can be swallowed into stomach, and you can see this stepwise increase of the accumulated swallowed drug amount, and also you have the amount of drug dissolved in the saliva will decrease. And overall, you can also using the regional absorption profile to get the prediction of drug absorbed via oral mucosa. Here in this plot, we predicted about 18%. of the Zolpidem was actually absorbed via oral mucosa. So this is the last slide for the case example, so we'll go to a summary. So here today we actually present 4 different cases. So case study 1 is the incorporation of the in vitro distribution data with the PBPK model to demonstrate a useful approach to access the proton pump inhibitor effect, which is also the effect of Achlorhydria or drug-drug interaction with acid-reducing agency, and on the clinical performance of various formulations. Case study 2, we also talk about the calibration of the absorption model on lower GI and how we're using this approach to support controlled release formulations in humans. Case study 3 is more like absorption PBPK model, a potential to complement the traditional IVIVC approach for the MR formulations. The last case example was mainly talk about novel physiological-based oral absorption for Zolpidem Sublingual tablets. So overall, I think actually for this presentation, we're trying to say the GastroPlus is a very useful software and also offer very good flexibility. So users can actually customize and refine the models, particularly as we see in the case study 1 and 3. And then we can use this GastroPlus model and be further extended to address various aspects of pharmaceutical and biopharmaceutical questions in the formulation development. So I will stop here, and then I will go into the Q&A section. And thanks a lot for the audience to listen to our presentation. And we will go ahead to answer the questions. Thank you.
Thank you very much, both Binfeng and Wei, for a very interesting presentation. We have had a number of questions, so I think we'll go to the questions now. Obviously, you presented on a number of case studies, and we've got questions on multiple case studies. So I think what I would propose to do is to start off with the first case study, which was the proton pump inhibitor, the PPI, and just run through the questions on that, and then we can go to the subsequent. So we had a question from Mohammed Shebli, or a couple of questions actually. He was asking which proton pump inhibitor was used in the clinical studies, and also his other question, the particular drug that was used, did it have any involvement of intestinal transporters or metabolism?
Okay. So, so the tablets that we presented in case study 1 was co-administered with a 40 milligram Protonix, which is pantoprazole, delayed release tablets on day 6 after multiple day dosing of the same formulation, and the dose is 40 milligrams. And regarding the question about the involvement of intestinal transporters or metabolism and how to handle in GastroPlus simulations, I guess in this case, since the PK is linear in the range of interest that we're looking at up to 40 milligram dose, well, up to 80 milligram dose. So I don't think that this is a real concern in this case, but certainly that if we have any of the transporter and metabolism involvement which alter the PK, for example, the overdose proportional PK, because of the saturation of the transporter and the metabolism, we can always incorporate it into the GastroPlus using the metabolism transporter module. So I think that one thing that I think is important is to look at the clinical PK data as much as possible. For example, first in human, the single ascending dose study, and to look at the linearity of the PK and to look at the food effect of the compound. I think these would certainly all be helpful for us to build up the GastroPlus model.
Thank you. So the next question came from Peng Zhu. and he was asking, is there a lag time between PK and PD of PPI?
I'm not sure if I understand the question correctly, but I guess that if the question is to ask if there's a lag time between PK and PD of the PPI, I think we can probably consider it as the regular case. Just if there's a lag time between the PK and PD, I think it's mostly compound dependent, case by case thing, and the PPI effect is mostly affecting, as we are looking at, is mostly affecting the absorption and the PK part of the behavior of the drug. So regarding the PK and PD delay lag time, I think it's a separate question. So I guess it's a case-by-case thing for different compounds.
Yeah, I mean, maybe another way of interpreting the question is, or another way of looking at it, was the PPI dosed before the other compound that was looked at? So did we allow time for the PPI to take effect?
Yes.
Was it like pre-dose?
I think they probably have at least 2 doses before they dose the compound, because PPI actually takes time to reach a steady state. I think it's 2 doses. One dose each before the dosing day, the night before the dosing day.
Yeah. So as I mentioned in the first question, so we co-dose the tablets with the 40 milligram delayed release protonics tablets. And on day 6, actually, after several days of the dosage of the same formulation, just to make sure that it is having effect of the PPI agent.
Yeah. Good. Okay. Another question from Mohammed Shebli. Is the colonic permeability calibration in dog compound dependent or a fixed physiological parameter?
Yeah, I think the question is, if I understand correctly, was trying to look at if the colonic permeability calibration in dogs, that when we adjust the colonic permeability ASF, if it's compound specific or if it's sort of a uniform adjustment. So I think that within our group, we have been looking at several cases using this method that I described in case study number 2. So I would say that the ASF value adjustment is certainly compound dependent because of the different physical chemical properties of the compound, and I think it is conceivable that it will be compound dependent ASF adjustment. But this method that I presented in case study number 2 is basically paved the way that how we adjusted based on the preclinical data and all the way to the clinical data, so that we can adjust the ASF settings accordingly.
Do you think moving from animal data to human data, that scaling that you did, do you think that's compound specific?
Yes. Yes, this, according to our experiences, yes, it is compound specific.
Okay, thank you. So the next question came from Janki Andaria, and the question is, how much difference between observed and predicted values are acceptable? So when we run a simulation and you compare the simulated with the observed, what do you look at for a reasonable prediction error?
Well, I think that it is not an easy question to answer, I guess. Well, I think all models are wrong. So I mean, some models are useful. I think we just, I think I would just emphasize the usefulness of the compound. So as long as that, for example, in the case study number 1, that even though that we have about 25% or to 30%-ish of the prediction error between the predicted outcome of the PPI effect versus the observed PPI effect in clinic, I would say still that it is at least pointing us to the right direction that we actually advanced our formulation, the enabling formulation to the clinic, and move the project forward. So I guess if we just look at the numbers, it's probably, you can see a difference here, but at least directional wise, I wouldn't say that this is a very big issue for us. And also we actually applied this model to other tested formulation too of this compound. But I didn't show the data here. But at the end, we basically advanced this compound, this formulation, as well as the other enabling formulation to the clinic. And we got the clinical data back, and we are satisfied, and team is also satisfied on the formulation development on this.
Okay. Later on in the presentation, you talked about IVIVC, and you highlighted where we had prediction errors generally below 15%. Where did that 15% cutoff come from?
So the IVIVC 15%, that deviation, actually I think is based on a successful criteria of IVIVC models. Concluded by, suggested by the regulatory agency guidance. So that's a little different because the case example 3, the IVIVC model is trying to make a successful IVIVC model. So it's more so this criteria will be more restricted, right? Because we want to make sure the IVIVC model will work not only by fitting the curve nicely, but also make sure those point estimates are within our prediction error, suggested by the guidance. So I would not say that we will always using 15% as a prediction cutoff accuracy, cutoff value for all different models. It's still case dependent.
Yeah, understood. So the next question was from Kailai Fong, and the question is, would you elaborate on the dog absorption model? In particular, was it chronically cannulated?
Okay, so for this one, so the dogs that we, the process that we were using, as far as I know, is that we dose the dogs colonically via a retrograde catheter method with our tested formulation, and goes all the way towards ascending colon. And then after the dose, we can have post-dose water flush with small volume. And then pretty much this is the way that we dose it to look at the sort of relative bioavailability of the chronic dosing. So pretty much we can use this method and compare it to the oral dosage of the same formulation to look at, to have some level of understanding of the potential of lower gut permeation of a certain compound.
Okay, thank you. And then we had a question on dissolution test condition. I think moving now towards the work we presented on the controlled release formulation. The question from Michihawa Kayama was, do you know suitable dissolution test conditions to fit GastroPlus? And he's saying when we try to fit CR formulation, it's difficult to know which dissolution condition is suitable to fit. So I think you're talking about the methods used in an in vitro dissolution test.
Yeah, I think this is a very good question, but also I think it's also difficult to answer because, it's probably difficult to say what is the best dissolution test conditions to use to fit GastroPlus, because that's the purpose of we need to do in vitro, in vivo correlation, right? Because if we're using in vitro profile, yes, fit the PK perfectly, we probably don't need to worry about the correlations. But for the particular case examples here, I think the dissolution method that we use is pretty standard. USP2 system, 100 RPM. Of course, this is very standard approach of the dissolution test. But I guess I cannot say I will say there's a perfect dissolution method you can use to get the CR formulations, right? Because I mean, of course, I think that's a step of keep refining the method and modeling. So it's kind of like keeping moving forward. I think that's probably the best answer I can give.
Yeah.
Yeah, I agree. I don't think that we can claim that we can predict. We use the certain dissolution method and test and to predict the absolute exposure of the CR formulation in GastroPlus. I don't think that anyone, at least at this stage, that we can claim that. But at least we can look at the relative exposure of different formulations. Let's say slow, medium, fast release formulations that we are investigating and give our feedback to the formulation team. And then on the other hand, that as Bingfen mentioned, that I think what we can do is also refine our model when we have the clinical data, and then I think this is always an evolving process. It's not like a standing model.
Yeah. And I think to some extent could well be dependent on the type of formulation that's being used as well. I think it's as you say, it's hard to actually assign a particular dissolution test that is best. Okay. Next question from Chakrada Lagishetty. Again around IVIVC. We can simulate in vivo PK profiles from in vitro release in GastroPlus. However, can we predict in vitro release profiles if in vivo PK profiles are known?
I mean, I
think you're saying, can we work back from in vivo PK profiles to get an in vitro release profile?
I think the GastroPlus model, what we can do, I mean, if you have the optimization model, what you can do is, if you have an in vivo PK profile, you can back calculate the in vivo release profile, not in vitro release profile, but in vivo release profile by optimizing the Weibull function. Because in vitro release profile depends on your experimental condition, right? But the in vivo release profile could be a target of your in vitro release study. So I think the GastroPlus model actually can get the prediction. I mean, get the if you use an optimization model, you can get a profile of in vivo release profile. That might be a target formulation. I mean, the target dissolution profile an in vitro study wants to achieve. So that's I think.
Yep. Yep. So it can be done, I think, is what you're saying. Is that right?
Yeah. I mean, I think I think based on this method, you can predict, you can get a targeted in vivo targeted in vitro release profile, which is the in vivo predicted profile. So whether, I mean, of course, next step is to go to switch that information back to the formulation or analytical team to see whether they can build the dissolution method to reach that in vivo profile.
Yeah. Okay, good. So we've now got a series of questions, and there seems to be quite a lot of interest around adjusting of the absorption scale factor in the controlled release models. So a number of questions around how was the optimization of the ASF done, where do those optimized values come from, do you need to optimize ASF for different formulations? So do you think you can just talk a little bit more around that, please?
Yeah, so the optimization done on ASF values, so I think both Wei and I are using a pretty similar approach. Basically, the purpose is trying to fit the pharmacokinetic profile, right, the concentration profile. So because you can imagine that the ASF value actually is just a multiplier. So the rate constant equals to the permeability, the effective permeability times the ASF values. And then you're using that numbers to calculate, to further calculate absorption rate. So the purpose of this is just trying to adjust ASF value to get absorption rate that can describe the PK profile. So in my case, example of that IVIVC models, I actually adjust the ASF value of each class compound simultaneously, which basically you can select if you are familiar with optimization models, basically you can select a multiple drug record. So we actually set a slow, intermediate, and fast record, and you can play around with optimization model to fit ASF value simultaneously. And you can, in the optimization model, you can also set some criteria. You can maybe a little bit overweight of the Cmax and AUC, because these two will be the key point estimate. And so by doing this, we do simultaneous optimization. But before that, I probably would suggest you to also use a manual optimization method. Just play around with this number to see which number is more close to the to fit the three different PK profile in each class of the formulations. Because it might be difficult to use the optimization model to directly search for right numbers. But with the manual optimization first, you will have a rough idea about what the range of this number will be. I think that's my strategy. Is anything Wei, you have your strategy you can share with?
Yeah, I think I just want to add on a little bit to that. So what Bingfen said was correct, that we can use the manual fitting at least to set up an initial value for the optimization, which certainly we don't want to get to the local minimum that might be deviated from the actual value, the correct value. So, and also what I want to add another thing is that I think that just towards the question of if we fit the ASF value of each GI segment, so I think I will just speak on behalf of my case, which is using the regional absorption data coming from the dogs. So in that case, actually we get rid of all other compartments from the small intestine, from stomach towards small intestine. So the only thing left is the colonic compartments, either one compartment model or two compartment model. So with that, it's kind of a more cleaner, model, because otherwise, if it is an oral dosage form, and I think this is very difficult to figure out the colonic contribution of the overall absorption, especially if the majority of the drug has already been absorbed in the small intestinal range.
Yeah. So one of the comments that came in from Peng Zhu was a note that there seemed to be, for each formulation, a different set of ASF. Was that the case? Did we find the need to alter ASF depending on the formulation? And then the follow-on question was, could that be related to dissolution rates?
So because I think if I understand the question correctly, they're asking, because we're using different sets of ASF value for each class of formulation, not each formulation, but class of formulation. So each class formulation, we have three different release rates. Because formulation is still in a different class, so I think their mechanism of release will be slightly different. Of course, ideally, if one set of ASF values can be used to predict the PK of different class of formulation correctly, that would be the ideal condition. But in most of the times, I mean, I also tried this approach, but basically it's not very successful. So you probably can predict the fast and intermediate rate for formulation more closely, but we see a pretty large deviation on slow release formulations when we're using. So typically the other way we try to using one asset the AS values. We fit one set of AS value with one class of formulation, and with this and we get this AS value, then we're using this set of AS values to predict another class of formulations. I mean, I think that's the ideal way we should do that, because that's I think in IVIVC criteria we call this something like internal validation and external validation. However, such approach is not very successful. I mean, at least for the slow release formulation at the other class of formulations. So in this case, we are kind of like still switch back to using different set of AS value for different formulations. So I think that probably currently that's the best approach we can do. So in the future, if there's some other release rate in that class formulation, we can use in the model to predict that. So I think that's the current the goal of this model. And I think Panto also asked about the question related to quantitative correlation between optimized ASF and the in vitro drug release parameter. We didn't check that actually, because I think the model itself is trying to make the IVIVC model correctly with some mechanism, but mainly still based on whether mathematically the model can be correct. We didn't really just, I think the ASF value is just a multiplier to the, but I think that's still a good suggestion. Maybe we can look into look into those correlations in the future.
Yeah, I think that's a very good suggestion. Yeah. And do you think if you if you have an in vitro and vivo correlation, for example, so your in vitro method really reflects the in vivo release, do you think that would help in these situations?
Yeah, if the correlation is better, of course, this will be much easier. So because when we try the deconvolution convolution approach, we don't have very linear correlations for the release. So we have to set up two different stages to get the model work for the which is deconvolution convolution model. So I mean, I think it's a little bit more complicated for people to use such kind of approach. So because we have the PBPK model at hand, right, and this just by adjusting the AS values as a multiplier, and you have more flexibility because you can easily kind of like adjust the AS values at different GS segments, and you have non-different compartment in your ACAT models, so you can adjust different values. Of course, we also don't want to make it too over-engineering, because that's why I want to set 5 AS values, but trying to reflect the differences between upgen and lower gen, and we found that insight, which is good. Okay.
Okay, good. We've got a question from Shilin Chen. For the case study compound B, can the adjusted ASF values for dogs apply directly to the human model?
Well, I think that if we apply this, so the model was set based on the ASF scalar. So obviously the default dog model, the ACAT model in GastroPlus, and the default human ACAT model in GastroPlus, the ASF settings are different, if not a large difference. So if we directly apply the dog ASF model to humans, I haven't tried that, but I think that it probably will not be the ideal situation to have a reasonable prediction. I mean, So the way that we have been doing here using the scalar, it's sort of like a ratio. Get out of the dog model is just trying to adjust the human model the same way as what we observed in dog model, which I think this is a more reasonable way to adjust the ASF value.
Yeah. Okay. She then also asked, did you have clinical data to verify your prediction of human in vivo PK for the 3 controlled release formulations?
Yeah, we do have the clinical data in the last slide of the case study number 2, but I just couldn't share this data here because of the confidential reason. The project is still active, so but I did mention that just from the AUC side, the prediction is largely in line with for the slow, medium, and fast release formulations, and then you can also see the differentiation between these 3 formulations in the predicted PK profile. Yeah.
And I think one last question to finish on, because we're very close to time. In terms of the risks, there are always simulations. How do you manage the risks in prediction? In terms of perhaps how you communicate the results.
Yeah. So I think the slides I show in the case sample, that slide here, the flow diagram of using a PBPK Physiologically-Based Absorption Model, which this flow diagram also published by Xingyuan Zhang, actually Dr. Xingyuan Zhang in 2011 ACAT journal. I think that diagram will give you a pretty good idea about how to control the risk of the model. So I think one thing is, if you think about the model optimization and verification, so in the middle of the box, that's probably a GastroPlus user should spend most of the time on to optimize the model. And then I also try to think how we can verify the model, validate the models. Should we try different dosing regimens? Does the model only works for one dosing regimen or only one formulation? But sometimes it will also have to be balanced between the models excellency versus practical situation, because sometimes we don't really have lots of data sets to work with, and of course, if you have less verification steps, basically the utility or the range of the utility will be relatively limited. So basically, this step is trying to ask the user to build a confidence interval of the model of yourself, and think about at this confidence interval how you use the model more wisely, or make the model more predictable, and make the model prediction more accurate. So I think that's my philosophy to control the risk of modeling. Maybe we can share here on that.
Yes, I think that we certainly it's an evolving process, and we are not stopping that. If there is any request from the formulation team, from the clinical team, that ask, there's anything that we can further optimize our model, I think it is certainly opportunity that for us, and also we also actively pursue these opportunities during the team discussions, and use the model simulation to help and guide the formulation development. And certainly there is a risk. I mean, if with limited data, we don't expect that the model to work all the time. It could be, when we get lucky, okay, so we can sort of fit the clinical data very well. But I think that at this time, what we can say is that we are just continuously optimizing our model with the data that feed in, and building our confidence, and make the model become more and more solid with more observed data validation.
Very good, thank you. So I think that's all of the questions that we've been through, and we've actually reached the end of our time for today as well. So I'd just like to finish by, again, thanking our two presenters today, Wei and Bingfeng, for excellent presentation and some very interesting discussion afterwards. So thank you very much to our two presenters. Very much appreciated that you've given your time and insight into this very interesting area. So thank you once again, and thank you to all the participants in the webinar, and we look forward to welcoming you on another webinar in the future. So thank you very much for now. Bye-bye.
Thank you. Bye.